Good morning and welcome to week 9 of AMP1. We are moving on from the skeletal system and beginning our exploration of the muscular system today. We have 3 different types of muscle tissue in our body. We're going to focus mainly on skeletal muscle today, but we'll also touch on smooth muscle and cardiac muscle, namely so we can be very clear about the differences and similarities between each type of tissue. Skeletal muscle functions to produce movement. It moves our bones, moves our joints, allows us to move through space, maintains the position of our body, so it keeps us upright, or if you want to do a plank, your muscle tissue keeps you in plank or a tree pose, whatever you're doing. Supports your soft tissues, guards your openings. We have sphincters that guard certain openings and circular muscles that say guard around your eyes, allow you to close and open your eyes, allow you to close and open your mouth. Maintains body temperature through shivering actions and provides a large nutrient reserve. Our skeletal muscles are made up of the muscle tissue. The cells of the muscle are called muscle fibers because they can be very long. Connective tissues which wrap around those muscle fibers, nerves that innervate the muscle fibers, and then blood vessels that supply the muscle fibers with blood. Our muscles are wrapped in 3 layers of connective tissue: the epimysium, the perimysium, and the endomysium. It's important to look at these words. Epi would be on the outside, peri would be in the middle, and endo would be on the inside. You can think of endothelium lining the blood vessels and the heart. So endomysium is the most inner lining of the muscles. So the epimysium is an exterior collagen layer. It's connected into the deep fascia and it surrounds the entire muscle. So it's really separating the muscle from the surrounding structures. So if we're looking at the biceps muscle, the epimysium would surround the entire bicep muscle. The perimysium is on the inside. So here, let's see. The epimysium would be on the outside like this. Oops. There we go. The perimysium is going to separate the muscle into individual fascicles, so bundles of muscle fibers. So if we were looking at the perimysium in this top picture, it would be the white area that is separating this into all of these different little bundles. That's the layer that contains the blood vessels and the nervous supply to the fascicles. So a fascicle is a bundle of individual muscle fibers. And then the endomysium surrounds those individual muscle fibers themselves. It has a capillary bed and nerve fibers that come into contact with the muscle cells. We're going to learn a lot about what happens at these neuromuscular junctions where the nerve and the muscle come together. And the endomysium contains myosatellite cells. So you're going to want to know that those are in the endomysium. Those are stem cells that divide and help to repair damaged muscle. Muscle is great in that it has the ability to repair, unlike the nervous system in many places. So they have these stem cells that are going to replace and repair damaged tissue, and those lie in the endomysium. So this is a great picture here that the epimysium surrounds the entire muscle, the perimysium surrounds the fascicle, and then the endomysium surrounds the individual muscle fiber. Our muscles are attached at either end. The endomysium, the perimysium, and the epimysium all come together at the ends of each end of the muscle, and they form our tendons or aponeurosis. So a tendon is like a thick bundle that attaches into a bone, and aponeurosis is like a sheet tendon that goes and attaches into the bone. So you have a lumbar aponeurosis, That's the sort of the main one. There's an aponeurosis that covers the back of your skull. So we have a few of those in the body. But that is the combination of the epimysium, the perimysium, and the endomysium all coming together. Skeletal muscles are voluntary. That means you have control over them. You can tell them to contract and you can tell them to relax. They're controlled by nerves of the central nervous system through the brain and spinal cord. And we're going to learn a lot more about that in 2 weeks, well, starting in 2 weeks. Blood vessels, we need a ton of blood supply to get to these muscles. Muscles need a lot of ATP for energy. ATP requires oxygen. Oxygen is carried in the blood. So we have a lot of blood coming in, bringing large amounts of oxygen, bringing nutrients, and then carrying away the waste products that are created during muscular contraction. Our muscle fibers are long, so our cells can be very long. They are developed through fusion of myoblasts. So myoblasts are sort of our early cells. You can see up here we've got some myoblasts, and they connect together and then end up fusing together to form these long cells. You can see that myoblasts have individual nuclei up here, and then as they fuse together, the nuclei all fuse into one cell. So skeletal muscle fibers are multinucleate because they contain the nuclei from all of the myoblasts that came together to form that cell. We also have those myosatellite cells that we talked about. They are myoblasts that did not fuse, so they remain in the system as a myosatellite cell, and those are taken care of and dividing to create additional muscle cells or to create additional repair when muscles are damaged. The cell membrane, so it's surrounded by the epimysium, but then we have a cell membrane like any other cell. A cell membrane is a very basic part of a cell. And so the cell membrane of a skeletal muscle cell is called the sarcolemma. So it's just a fancy term for cell membrane of a muscle cell. The sarcolemma surrounds the sarcoplasm, which is the cytoplasm of the cell. And a change in transmembrane potential is what begins our contractions. We're going to talk more about that later, but remember from week 2, I believe, we talked about positive charges on one side of a plasma membrane and negative charges on the other side of a plasma membrane, and how they're separated. The charges are separated by the plasma membrane, which gives a concentration gradient, and a change in that concentration gradient excites that membrane. Within our muscle cells, we have what are called T-tubules. These are extensions of the sarcolemma. They're continuous with the surface of the sarcolemma, and they dive deep down through the sarcoplasm. These are what transmit the action potential that the cell membrane generates. They transmit that action potential throughout the cell and allow the entire muscle to contract at the same time. Myofibrils are lengthwise subdivisions within a muscle fiber. They are made up of bundles of protein filaments, which are called myofilaments. The main myofilaments are actin and myosin. Actin is the thin filament, and myosin is the thick filament. The interaction of actin and myosin is what creates contraction of the muscles. So here you can see a myofibril. Our skeletal muscles are striped. They have a striped pattern because they have alternating layers of thick filaments and thin filaments. The bands, let's see, in my additional handout that is posted on your website this week, there's one about sarcomeres. The sarcomere is that actual repeating unit of the myofibrils. It is the smallest functional unit of a muscle fiber, and it's divided into multiple regions. In another couple of slides, I'm going to go into more detail in those regions. But for now, the thick filaments make up the A band, and the thin filaments make up the I band. So the sarcoplasmic reticulum is the membrane that surrounds each myofibril. It transmits the action potential to the myofibril, and it's stimulated by the action potential coming down the T-tubule. So in this picture here, you can see the sarcoplasmic reticulum is shown in blue. So it is really just enveloping all around those myofibrils. It's similar to smooth endoplasmic reticulum, and it functions to store calcium. That's the whole purpose of the sarcoplasmic reticulum, is calcium storage. It has these areas, let's see if I can draw them small here. So this area here on either side of that yellow, there's two terminal cistern or cisterna. And what those are expanded chambers where calcium is really being stored. And then the yellow that's surrounding the center of those two terminal cisterns is the T-tubule. So the combination of one T-tubule with two terminal cisterns right there is called a triad. Here we go. So the triad, one T-tubule, two terminal cisterns. The terminal cisterns are going to concentrate the calcium through calcium ion pumps, which require ATP, and then they're going to release calcium when the action potential arrives, and the calcium is what triggers muscle contraction. So there's always calcium in the system. When the T-tubule transmits an action potential, the terminal cistern is going to release its calcium. That's going to interact in a cascade that we're going to learn in a minute. But when muscle contraction is over, that calcium will get pumped back into the T-tubules via calcium pumps, which require ATP. So here's our sarcomere. So the sarcomere is the contractile unit of the muscle, and it is bands of actin and myosin, or thick filaments and thin filaments are actin, thick filaments are myosin. And this is what gives us that striped pattern to our muscle. So this is where you're going to want to look at those extra couple of handouts that I gave you. The A band, which you can see here, is this whole area that contains all of the thick filaments. So from one tip of thick filament to the other tip of thick filament is the A band. So the A band contains thick and thin filaments, but it runs the length of the thick filaments. The H band right here in the center only contains thick filaments. So it's very small, and it's only showing, you can see the red is the actin, and this dark blue purple color is the myosin. So where the actin stops on either side, there's a gap, so that in this very central region, you only see myosin, and so that is the H band, where there is only myosin, no actin. Next is the M line. So the M line is literally this line right in the middle of the sarcomere. So it's where the two layers of myosin connect together in the center. So it's the center line of the sarcomere is the M line. Here we have the zone of overlap. That is where you have overlap between the actin in the red and the myosin in the purple. I'm just going to call that purple. So the zone of overlap is where you have actin and myosin overlapping one another. So the A band consists of the H band, the M line, and the zone of overlap. So all of that is within the A band. And two more. So next we have the I band. The I band is with only thin filaments. So the I band is here from the edge of the thick filaments of one sarcomere to the edge of the thick filaments of the next sarcomere. So the I band spans two portions of two sarcomeres. And in the center of the I band here is the Z line. It's shown over here. So the Z line is where the actin comes together with the actin of the next sarcomere. So a sarcomere runs from Z line to Z line. And the Z lines are in the center of the I bands. The last thing you want to know on here is if you see these red lines, the purple lines, the myosin stop here. They're stopping. And then you see this green squiggly line that continues after the myosin has stopped. That is titin, which you can see up here. Titin. Sorry, hold on. There we go. Titin. So titin is an elastic filament, and what that does is it comes in and attaches to the Z line, and it really just helps to stabilize the myosin. So the M line is the center of the A band at the very midline of the sarcomere. The Z lines are at the centers of the I band, and they mark either end of the sarcomere. So sarcomere goes Z line to Z line. The M line is in the center. The zone of overlap is where those thick and thin filaments are overlapping. The H band is the area around the M line that only has thick filaments, no thin filaments. And then the titin is those elastic protein that are reaching from the tips of the thick filaments out to the Z line and stabilizing those thick filaments in place. This slide goes back to look at those connective tissue coverings again, but I think it's a nice slide. It's on page 299 in your textbook. So it's showing you that the entire skeletal muscle is surrounded by epimysium and contains fascicles. So these are all fascicles which are bundles of muscle fibers. The second one is a fascicle. So the fascicle is surrounded by the perimysium, and it contains individual muscle fibers. And then the individual muscle fibers are surrounded by the endomysium, and they contain myofibrils. So the sarcomere function is for muscle contraction. So we have the triad that we talked about before, which is the T-tubule in the center with one terminal cistern on either side. Calcium gets released from that terminal cistern, and that's what initiates the contraction of skeletal muscle. And the contraction is the interaction of actin and myosin together. Our actin, don't worry about this F and G actin. I will not ask you to delineate between the two. So actin is a twisted strand of globular molecules, and you can see down here that all of these big globs are actin molecules that I'm coloring in yellow here. On each actin, there is what's called an active site. So here you can see this active site is the yellow. The orange would be the rest of the globular protein, but the yellow is the active site. And the active site is where myosin can bind to the actin. That's also called the myosin binding site. So the myosin head can bind to the actin there and induce contraction. Tropomyosin is a double strand. So it's in blue down here. This is tropomyosin, and the tropomyosin kind of wraps around the actin, and it covers up the active sites on the actin. So our tropomyosin wraps around the actin and covers up those active sites. And the tropomyosin is held in place by troponin. And troponin is a protein that has 3 subunits. So when we do this in lecture, we usually take Play-Doh and we create a line of actin, and then we cover it up with a thin line of tropomyosin, and then we make 3 little balls into troponin, and the troponin sits there and it holds the tropomyosin onto the actin, covering up those active sites. So when tropomyosin is covering the active sites, muscle contraction cannot occur. The troponin, one of those 3 subunits, binds to the tropomyosin. The second one binds to the actin to hold the tropomyosin and the troponin in place to the actin. And then the third one is a free receptor, and it is sensitive to calcium. It's a calcium receptor. So what happens to actually create contraction is you have calcium coming in. So calcium gets released from the sarcoplasmic reticulum. And so let's say calcium comes in here. The calcium goes and binds to the troponin and creates this, so it binds to the troponin-tropomyosin complex. So that's basically just saying that the troponin and the tropomyosin are bound together. So the calcium comes in, binds to troponin, and when that binding happens, it causes the troponin to change its shape. So the troponin rolls over, and when it does so, it rolls the tropomyosin off of the active sites on the actin so that it exposes those active sites. And that's what allows the myosin heads to bind to the actin and cause a muscle contraction. So to say that one more time, initiation of muscle contraction requires the release of calcium from the sarcoplasmic reticulum. Calcium will bind to the troponin, change the shape of the troponin, which then pulls the tropomyosin off of the active sites on the actin, therefore allowing the myosin to bind to the actin. Our thick filaments are our myosin filaments. They are made up of a head and a tail, which you can see over here. The tails all intertwine together. So they end up looking like this up here, where you have all of these tails together, and then you have the myosin heads just kind of sticking off of the tail like that. During contraction, the myosin heads are going to bind to the actin, forming what we call a cross bridge. And then the heads are going to pivot and pull the actin closer to the M line. So this whole cycle is called the sliding filament theory. The sliding filament theory is that the thin filaments of actin are going to get pulled by the myosin heads, and they're going to slide toward the M line along the thick filament. The width of the A band stays the same because the myosin length does not change. The Z lines, however, move closer together as the actin is being pulled in and the titin is going to get compressed. So here's our picture. On the right-hand side, we have a sarcomere at rest. And so we can see that there's this big H band in the center where there's only myosin, no actin. And so this is at rest. And then over on the right-hand side, we can see that the actin has been pulled inward. So the H band is much smaller, and the titin, which was long on this side is now compressed. So the actin is the same length, the myosin is the same length, but the actual actin fibers are sliding along the myosin fibers so that they are coming closer to the M line and shortening that muscle. This is the sliding filament theory. So we just went over in my additional notes, that was all page 2. So where it says contraction cycle. So the contraction cycle starts with calcium binding to troponin. The troponin changes the position of the tropomyosin, uncovers the active site on the actin molecule. The myosin heads then bind to the active site and form a cross bridge. Then the myosin heads are going to pivot toward the M line. This is called the power stroke, and that's going to shorten the sarcomere as the actin moves along the myosin. Then the myosin head is going to detach and reactivate. So we're going to get to those last couple bits in a minute. But that page 2 of the contraction cycle is what happens within the individual muscle cell. Now we're going to move on to, we started with calcium needs to get released. So now we need to figure out how and why does that calcium get released to stimulate muscle contraction. So we have a nerve that stimulates the sarcolemma, or the cell membrane of the muscle fiber, causing what's called excitation-contraction coupling. So the nerve impulse is going to excite or depolarize that sarcolemma, that cell membrane, and that is then going to lead to contraction occurring. So we have excitation first and then contraction. So the sarcoplasmic reticulum is going to release calcium, triggering the interactions between the thick and thin filaments, consuming ATP and producing tension. So tension is what happens when you shorten a muscle fiber from either side. That produces, or when you're pulling from either side. It doesn't necessarily have to shorten, but when you're attempting to shorten, you're increasing tension. So let's look at what happens at the neuromuscular junction. The neuromuscular junction is where the nerve comes and synapses upon the muscle. So that's our neuromuscular junction. The action potential is going to travel along the nerve fiber until it reaches the synaptic terminal. So the synaptic terminal is where the nerve fibers come in and synapse upon the sarcolemma. At the synaptic terminal, we have a neurotransmitter that gets released, and that neurotransmitter is acetylcholine. So the acetylcholine gets released from the nerve itself into what's called the synaptic cleft, which is just the space between the neuron and the muscle fiber. There's a little miniature space in between there, so that's the synaptic cleft. And this whole part here is called the motor end plate. So the motor end plate is that part of the muscle where the neuron is synapsing. So we have an action potential travels down the nerve, triggering the release of acetylcholine. That's where we're at so far. Acetylcholine travels across the synaptic cleft and binds to the membrane receptors on the sarcolemma. So there are receptors, acetylcholine receptors on the cell membrane, the sarcolemma surrounding that skeletal muscle fiber. Acetylcholine binds to them, and what happens when the acetylcholine binds is it causes a depolarization, which causes the sodium gates to open, and then sodium rushes into the sarcoplasm. So there's always sodium hanging out in the interstitial fluid surrounding the muscle cells, but it can't come through because of the sarcolemma is not permeable to sodium ions. So when the acetylcholine binds, that triggers the sarcolemma to open its calcium channel gates, and then, its sodium channel gates, and then sodium rushes in to the sarcolemma. Any additional acetylcholine that is left in the synaptic cleft that did not bind to the sarcolemma is then broken down by an enzyme called acetylcholine esterase. So here it is step by step. So in this first picture, we're looking at the nerve here. This is the synaptic cleft, and this would be the motor end plate. So this is the motor end plate is the sarcolemma. So first things first, the action potential is going to arrive at the end of the neuron, and that action potential will trigger the release of acetylcholine. I guess we're just starting with the action potential arrives. And these are vesicles of acetylcholine, which is how our neurotransmitters get released. We're going to learn a lot more about that when we get into the nervous system. So step 2, the action potential triggers the neuron to release acetylcholine into the synaptic cleft. The acetylcholine is then going to bind to the receptors on the sarcolemma, and that binding is going to then cause sodium ions to rush into the sarcoplasm. When sodium rushes into the sarcoplasm, it creates a second action potential along the sarcolemma. So we have two action potentials involved here. The first action potential comes through the nerve, triggers the release of acetylcholine. Acetylcholine crosses the synaptic cleft. It binds to the sarcolemma, and it triggers the opening of sodium channel gates. The sodium then rushes into the sarcoplasm, and that triggers a second action potential which travels along the whole entire sarcolemma. So it's called an excitable membrane, which means that it can carry an electrical current, and that's what an action potential is. It's a large electrical current. In this picture over here, you can see these little yellow moons are the acetylcholine esterase, which is in there cleaning up the additional acetylcholine. So once this second action potential occurs, then we go back to neutral, sort of normal, and nothing is happening at that neuromuscular junction until you have another action potential that comes in. So the action potential is generated by an increase in sodium ions through the sarcolemma into the sarcoplasm. It travels along the entire sarcolemma, so it travels down the T-tubules because they are extensions of the sarcolemma deep into the sarcoplasm, and the T-tubule is wrapped around the sarcoplasmic reticulum at the triad. So the excitation is that action potential reaching the triad, and when that action potential reaches the triad, it triggers the sarcoplasmic reticulum to release its calcium. Then the release of calcium is what the calcium binds to the troponin, moves the tropomyosin, uncovers the active sites on the actin, allows the myosin heads to bind to the actin, and then the power stroke is the actual contraction. So the release of calcium from the sarcoplasmic reticulum triggers muscle contraction. That muscle contraction requires the myosin heads to be in what's called a cocked position, where they are loaded by the energy from an ATP molecule. So the piece that we just finished was the excitation of getting the action potential to travel along the sarcolemma down the T-tubule and stimulate the release of calcium from the terminal cistern. So now we're going to look at the actual contraction again. So with contraction, it begins with the arrival of calcium. Here's our calcium. And the calcium is going to bind to the troponin. So you see on this left-hand side, the tropomyosin, which is this nice blue line. The tropomyosin is covering the active sites on the actin. So then when we move over to the other side, this calcium is binding here. That pulls the tropomyosin off of the active sites and exposes those active sites on the actin. Once those active sites are uncovered, the myosin head is going to bind to that actin. Now, the myosin has already interacted with an ATP molecule, and it has split that ATP into an ADP and a phosphate. So that means that myosin head is in that cocked position. So let's do this. All right, so the myosin is holding the ADP is like the potential energy from the ATP. So the release of ADP would release a whole bunch of energy. So the myosin head is cocked. It binds with the actin at the active site, and then it releases its ADP and uses that energy to pull the actin forward. So now the whole actin molecule or actin strand is being pulled toward the M line. Now we have to detach the myosin from the actin. And the way to detach the myosin head from the actin is by binding another molecule of ATP. So the ATP binds to the myosin head, and the myosin head is going to split that ATP into another ADP and phosphate and recock and get ready to bind again. So this is a repeated process of bind, pull, unbind, split the ATP, bind, pull, unbind, split the ATP. So every time it binds, it releases its ADP and phosphate while it's pulling the myosin toward the M line, and then it takes another ATP to bind to the myosin head to get it to release the actin and recock the myosin head. This is shown pretty nicely in the crash course video on muscle contraction, so I would definitely encourage you to go and watch those 2 to 12 times this week, because this is a lot of stuff to sort of keep in mind. Looking at my flow sheets would be really helpful of what happens in what order and how does this all occur. When you're looking at my written handouts, the muscle contraction and contraction cycle. Muscle contraction says page 1, contraction cycle says page 2. So the muscle contraction starts at the action potential arriving at the axon terminal. So the axon terminal is the tip of the nerve that stimulates the release of ACH, and it gets released through exocytosis because it's in those vesicles. The acetylcholine binds to the receptors on the sarcolemma, and that binding opens the sodium gates and increases the sodium concentration in the sarcoplasm. That creates a second action potential along the sarcolemma. That action potential travels down the T-tubule, reaches the triad, stimulates the release of calcium from the sarcoplasmic reticulum, and that calcium triggers the beginning of the contraction of the myofibril. Then you can go on to page 2, which is the one we already gone over, where the calcium that gets released then binds to troponin, moves the tropomyosin, uncovers the active sites on the actin. The myosin head then binds, releases its ADP and phosphate while it pivots and pulls the actin toward the M line. Then it's going to bind a second ATP so it can release the actin, split that ATP into an ADP and phosphate and recock and get ready to bind again. The shortening of a muscle fiber occurs as the sarcomeres are going to shorten. So you can see on the top here, we've got this long muscle fiber, and the thick bands and the thin bands, or the dark bands and the lighter bands, are both relatively wide. And then in the second one where the muscle is contracted, you can see that the I bands are much smaller, but the A bands remain the same length. So the actin is sliding over the myosin and shortening the muscle fiber. that shortening of the muscle fiber produces tension. The duration of the contraction, so how long that contraction lasts, depends on the neural stimulus. So is it a sustained stimulus? Is it repeated stimuli? It depends on how much calcium is in the sarcoplasm. If you need to have calcium in the sarcoplasm in order to have contraction. And it depends on the ability of ATP. We need ATP in order for the myosin heads to pivot. We also need ATP in order to pump the calcium back into the sarcoplasmic reticulum so that it can be released again. Once our contraction has completed, our muscles need to relax again. So during relaxation, calcium concentrations fall as the calcium pumps the calcium back into the sarcoplasmic reticulum with using ATP. The calcium detaches from the troponin as the concentrations are falling and gets pumped back into the sarcoplasmic reticulum. Without calcium, the troponin is going to change shape again and pull the tropomyosin back over the active sites on the actin to cover them up. But the sarcomeres remain contracted until there are outside forces that act upon them to stretch them back out. Rigor mortis is what happens after death when your muscles contract and they stay contracted. What happens is that the ion pumps, the calcium ion pumps, cease to function because when you die, you stop producing ATP. So the calcium cannot get pumped back into the sarcoplasmic reticulum, so it builds up in the sarcoplasm and creates this mass contraction that does not let go. On the left-hand side of this picture, we have the steps in muscle contraction. So we are starting up here with the release of acetylcholine at the synaptic terminal. That's going to trigger an action potential to come down the T-tubule and stimulate the release of calcium from the sarcoplasmic reticulum. Calcium binds to the troponin, removes the tropomyosin, uncovers the active sites on the actin, allows the myosin heads to bind, and then that binding of the myosin heads, the myosin will release its ADP and phosphate and pull the actin forward, thus shortening the muscle. For relaxation, the acetylcholine gets broken down by acetylcholine esterase, so we have no more stimulation coming in. So the calcium gets pumped back into the sarcoplasmic reticulum. The tropomyosin recovers the active sites on the actin when calcium unbinds from troponin. So the myosin relaxes because it can't continue to bind without more ATP and without the calcium moving the troponin. And so that the contraction ends, but it stays in a shortened state until you have passive return to its resting length. So we're going to talk about in a couple minutes, we'll talk about a few of the things that occur for muscles to relax and return back to their original length. So we're going to talk a little bit about tension. As a whole, a muscle fiber is either contracted or relaxed. You don't have a partial contraction of a single muscle cell. It's either the whole thing is going to contract or the whole thing is going to relax. Tension in a single muscle fiber depends on the number of cross bridges. So the cross bridge is where the myosin grabs onto the actin. So if you've got 10 cross bridges, it'll be a different end product than if you have 1,000 cross bridges. The fiber's resting length at the time of stimulation matters, and the frequency of stimulation matters. So in a single muscle fiber, we have the number of cross bridges depends on how much overlap there is between thick and thin filaments. So I want you to use your arms and consider that your arms are down at your sides. Somebody puts a 50-pound weight in your hand and asks you to pull that 50-pound weight, to do a bicep curl and pull it up to your shoulder. When your arm is fully straightened out, you have minimal contact between your actin and myosin and your biceps because your biceps is as long as it can possibly get. So you have fewer cross bridges to initiate that contraction. So it feels challenging to lift 50 pounds with a straight arm and do a bicep curl. Now put your elbow at a 90-degree angle. So as if you're standing up and you're trying to hold a tray. When somebody puts a 50-pound weight in your hand and you're at that 90-degree angle, you have twice as many, if not more, cross bridges than you did when your arm was straight and your muscle was lengthened. So from there, it's considerably easier to curl that 50 pounds, 45 degrees from from the 90 degrees up to getting that weight up to your shoulder. If you have your arms up at, instead of 90 degrees, say 120-degree angle, there's not a lot more that you can do to contract, to shorten that muscle, because your muscle is already almost as short as it gets. So that's what we mean by the number of cross bridges, is how much overlap is there between the thick filaments and the thin filaments. And there's kind of a sweet spot that is that is optimal, and that optimal is between 75 and 130% of that optimal length is where our normal resting sarcomere length is. Frequency of stimulation is how many nerve impulses are coming down to that muscle fiber in a row. So a single nerve stimulation is going to cause a single contraction, which is called a twitch. A muscle twitch lasts about 7 to 100 milliseconds. So that is super, super fast. And if you've ever had your eyelid that's going kind of crazy and it's contracting and twitching, those are single individual neural stimulations where you just get that twitch in your eyelid. Sustained muscular contractions, like if somebody gives you that 50 pounds and you're trying to hold it up and pull it up, that requires a lot of input from your nervous system to repeatedly tell you keep going, keep going, keep going, keep going. If you just had one single stimulus, you'd hold it and then you would drop it immediately. There's 3 phases that happen in the production of a twitch. So with this, we're looking at an individual muscle twitch. So there's a period before contraction called the latent period, where the action potential is moving through the sarcolemma and beginning to cause that release calcium. So until the sarcoplasmic reticulum releases calcium, nothing's really happening. And so that's our latent period right here. The next phase is the contraction phase. So with the contraction phase, we are building our calcium. So calcium is going up, it's binding, and we're building our muscle up to peak tension. There's a ton of calcium and all of the available cross bridges are happening. And then during the relaxation phase, calcium levels are falling and the active sites are getting recovered and the myosin is no longer binding, and so the tension is decreasing in that muscle. TREP, or TREPE, we have yet to figure out exactly how you pronounce that, is a stair-step increase in twitch tension. So what that means is you have a repeated stimulus, so the nerve is repeatedly telling you to contract, and that stimulus comes immediately after the relaxation phase. So if you relax and then you get another stimulus, you're going to make a slightly bigger tension the second time. And then another stimulus, another slightly bigger tension. So it causes a series of contractions with increasing tension. So in TREPE here, so this is TREPE or TREPE, whatever. So you would start with a small contraction and you slowly go up to what's considered maximal contraction, which is when you're getting the stimulus after the relaxation phase. This is the maximum amount of contraction that you can actually make. Wave summation occurs when you are getting repeated stimuli before the end of the relaxation phase. So your muscle's contracted, and before it's relaxed, it's getting stimulated again. And so that makes a more forceful twitch and a more forceful twitch every time that happens. So instead of the slow rise with that kind of max out here, wave summation is going to give you a steeper rise of more and more and more stronger twitches. All right, sorry about that. I had to get resituated with my notes. So incomplete tetanus happens when there is still a slight relaxation phase between each stimulus. So you can see here you're increasing in your tension, but then you kind of hit this maximum tension up there where you still have the slight relaxation phase in between stimuli. Complete tetanus occurs when there is no relaxation phase. You are just in complete contraction all the time. That happens, most people when they hear the word tetanus, they think of the disease where you step on a rusty nail and you get lockjaw and your muscles can't relax. That's a terrible thing. That's a pathological thing. But complete tetanus also happens when we are performing maximal capacity weightlifting. So if you're a weightlifter and you are bench pressing, I don't know, 500 pounds because you're some giant dude, then you have this maximal sustained contraction that keeps you from dropping that weight back onto your chest and killing yourself. So this does happen naturally, but it is not a regular occurrence. Interestingly, you'll learn more in AMP2 about this, but cardiac muscle cells cannot go into complete tetanus, which is very important. So a motor unit is a one nerve that's supplying hundreds of individual muscle fibers at the same time. So you have one nerve impulse and it creates all of these muscle fibers contracting at the same time, so they contract as a single unit. While this is happening, we have what's called multiple motor unit summation. So when you have a large muscle and it's got multiple motor units, one motor unit is going to contract all as one. The second motor unit will contract as one, the third, etc. So in order for your muscles to be smooth, they can't sustain continuous contraction. So you have this rotating of motor units. So the first motor unit is going to contract. And then while it's getting tired, the second motor unit is going to contract. And the first one's going to start to relax. And then the third one's going to kick in and allow the second one to relax. And then the first one's going to kick back in. So it's the motor units are getting stimulated slightly off from one another so that you can have smooth motion without exhausting any one motor unit in particular. Maximum tension is achieved when all motor units reach tetanus, and that is the one that you can only sustain for a very short period of time. Sustained tension versus maximum tension. Sustained tension is where you have a less than maximal tension, so it's still allowing motor units to rest slightly in rotation, and so you can sustain tension for a longer period of time. Muscle tone is the normal tension and firmness of a muscle at rest. And we know that we can change our muscle tone, our resting muscle tone, through exercise. The more you utilize those muscles, the greater their resting tone is going to be. Muscle units actively maintain our body position without motion, so they have some contraction to keep your head up. Your muscles in your neck have to be contracted to keep your head from just flopping over to the side. When muscle tone is increased, it increases your metabolic activity because it requires additional ATP. Because muscle tone requires some bit of muscle contraction, and when your muscles are contracted, they're utilizing ATP. So the greater your muscle tone, the greater your resting metabolic rate. So when you build muscle, you burn fat and sugars and all of those things more effectively. We have two types of skeletal muscle tension, then like muscle contractions that we're going to discuss. Isotonic contractions and isometric contractions. So isotonic means that the skeletal muscle actually changes in length, which results in motion. So here on the left, you can see this is an elongated muscle and the 2 kilogram weight is down on the table. Now we have lifted the weight up and actually shortened the muscle and created movement. So that is an isotonic contraction. If the muscle tension is greater than the load, you get a concentric contraction, which means the muscle gets shorter. So if you have a 5-pound weight in your hand and you contract your biceps and pull that weight up to your shoulder, that is a concentric contraction. Your biceps has shortened, your forearm has come closer to your shoulder, flexion has occurred. That is a concentric contraction. If, on the other hand, muscle tension is less than the load, so say you put a 100-pound weight in your hand and your elbows at 90 degrees, and that weight is too heavy for you to lift, so you're still contracting your muscle, but your muscle is lengthening at the same time, then that is called an eccentric contraction. Both of those are important in building muscle, so they can both be very healthy, but a concentric contraction shortens the muscle, and an eccentric contraction, the muscle is contracting, but it's also lengthening because the weight is too great for the ability of the muscle to maintain and manage that load. Isometric contractions mean that the length of the muscle does not change. But you're still developing tension within that muscle. So on the left side, you can see that this muscle is lengthened and sort of normal shaped. On this side, this muscle was unable to lift that weight, but you can see that it's getting thicker, so it is increasing its tension, but it's not actually shortening or lengthening. So the length is staying the same, but the tension in the muscle is growing. Resistance and the speed of contraction are inversely related. What this means is that the heavier the load, so the load would be the resistance, the longer it takes for the shortening to begin. So if you, again, have your arm in that 90-degree angle, 5 pounds, easier to lift, less resistance. 50 pounds, much harder to lift. It takes longer to initiate that contraction and the shortening of the muscle. After contraction, a muscle can return to resting length through elastic forces, which would be the elastic recoil of the muscle fibers. The titin is an elastic fiber. There's some elasticity in the surroundings of the muscle. Opposing muscle contractions. So if you contract your biceps to flex your arm, you could then contract your triceps, and that would lengthen your biceps and straighten your arm. And then gravity. If you contract your biceps and then you decide to just let it go, gravity is going to drop your arm back down and re-lengthen that biceps muscle for you. So this is just showing that if you have a heavy load, so a heavy resistance, it takes longer to begin shortening, and the smaller load, so the large load is down here being shown there, so it takes longer to shorten and it shortens less. Here's a medium-sized load, and then a small load you can shorten really quickly and move it really far and then drop back down. After contraction, so we already talked about this, elastic forces are the pull of the elastic ligaments, like or elastic elements, tendons and ligaments, which expand the sarcomere back to resting length. Opposing muscle contractions we just discussed with the biceps and the triceps, and gravity can take the place of opposing muscle contraction like we just discussed. Muscle contraction uses a lot of ATP, and especially sustained contraction. So if we're trying to go and work out and we're wanting to lift weight, we are going to need a ton of ATP. Our muscles, unfortunately, only store enough ATP to initiate a contraction, not to sustain contractions. So if you are going to want to sustain contractions, your muscle fibers are going to have to generate more ATP. ATP is adenosine triphosphate. We also have creatine phosphate, which is a molecule that can store additional ATP within a resting muscle. So when we're contracting our muscles, creatine phosphate can be broken down via the enzyme creatine phosphokinase. Anytime it says an ASE at the end, that is traditionally an enzyme. Enzymes, their whole job is to break down other things. And so the creatine phosphokinase breaks down the creatine phosphate and can release or generate additional ATP. ATP can be generated in two ways within our body, through aerobic metabolism and through anaerobic metabolism. Aerobic metabolism utilizes oxygen. It breaks down fatty acids and glucose, and it produces 34 molecules of ATP per 1 molecule of glucose that gets broken down. This is the primary source of energy for resting muscles. So if you're just kind of hanging out, aerobic metabolism all the time. And even during, if you do some light activity, like you're on the treadmill and you're walking, you're not like running full bore, but you're walking or, walking a little bit of a better pace, you're still doing all of that through aerobic metabolism. You may have heard a lot of exercises are aerobic exercises, or your mom might have said, I'm going to aerobics class, and that means that you're doing things at sort of minimal capacity, utilizing aerobic metabolism. So you're using oxygen to produce 34 ATP per 1 molecule of glucose. Anaerobic glycolysis is the opposite. It does not use oxygen, and it is primarily how we create ATP during peak muscular activity when our oxygen is at a minimum. And you'll notice that aerobic metabolism produces 34 ATP per molecule of glucose. Anaerobic metabolism produces 2 ATP per molecule of glucose. So it is much less efficient than aerobic, but it can be performed in the absence of oxygen. So it's breaking down glucose from glycogen, which gets stored in the skeletal muscles. So that's important for maximal contraction, high energy, sustained workouts, is anaerobic glycolysis. So this is on page 321 of your textbook, and it's just going over the different types of metabolism. So in our resting muscle, this is what we're starting with up here, our resting muscle, we're breaking down fatty acids and glucose, and we are creating ATP, and then our additional ATP is creating creatine phosphate, which is going to store energy for use later. With moderate activity, we break down, glycogen gets broken down to glucose, and then we create 2 ATP, and then we can also create ATP from ADP stores. It can get created into ATP. And then during peak activity, most ATP is produced through glycolysis. So we're making these 2 ATP. We're breaking down our creatine phosphate into ATP over here, but this is a much less efficient way to metabolize. So here we're creating 2 ATP per glucose. Over here we are creating 34 ATP per molecule of glucose. At peak exertion, our muscles don't have any oxygen to support the mitochondria, so they're relying on glycolysis for ATP. What happens during glycolysis is we build up pyruvic acid, and that pyruvic acid gets converted into lactic acid. When muscles can no longer perform, they are what we call fatigued. So muscle fatigue results when we deplete our metabolic reserves. So we have no more ATP, no more glucose, nothing else to break down. When we damage our sarcolemma or our sarcoplasmic reticulum, so we can't transmit the action potential with a damaged sarcolemma, we can't release calcium with a damaged sarcoplasmic reticulum. When our pH is low, so we have high levels of lactic acid, we have a low pH, that makes it more difficult to produce ATP, and we end up with exhaustion where we cannot physically move the way that we want to, and then it becomes very painful. Recovery period is the time required for exertion, after exertion, for muscles to return to normal. After moderate activity, it takes several hours to recover fully. After sustained higher activity, it can take up to a week to fully recover. So you want to be careful that you're not doing sustained high activities on a daily basis, because your body's never going to have time to fully recover. During recovery, oxygen becomes available again, and our mitochondria are going to be able to create ATP again. The Cori cycle is the removal of lactic acid by the liver. The liver converts that lactic acid into pyruvic acid, and that pyruvic acid can be broken down into glucose, which can get released to then recharge the glycogen reserves in the muscle. Remember that glycogen reserves are broken down in anaerobic metabolism. After exercise or large exertion, the body requires more oxygen to normalize the metabolic activities. And so the way that we get more oxygen is by increasing our pulse and increasing our respiratory rate. When we breathe more heavily and more quickly, we're bringing more oxygen into the lungs. The pulse speeds up, so it's delivering oxygenated blood more quickly to the tissues. And so that is counteracting the oxygen debt that we end up with after exercise. At rest, skeletal muscles are metabolizing fatty acids and storing glycogen, so they're taking glucose and making it into glycogen. During light activity, muscles are generating ATP through aerobic breakdown of carbohydrates, lipids, and amino acids. And at peak activity, energy is provided by anaerobic reactions and then generating lactic acid as a byproduct. Active muscles are producing heat. Up to 70% of muscle energy can be lost as heat, thus raising the body temperature. So when you exercise, you get beet red. Part of that is that heat loss. You're sweating, you're losing that heat that your muscles are producing. Hormones involved in muscle metabolism. Growth hormone stimulates the production of actin and myosin and increases the size of skeletal muscle, along with testosterone. Those two have the same effects there. Thyroid hormone increases the rate of energy consumption at rest. And epinephrine increases metabolism and increases the duration of stimulation and the force of contraction. So epinephrine, that's what gets released when you're in that fight or flight, and you can perform these Herculean activities, like, your kid gets pinned under a car and you can suddenly lift up that car. That's the release of epinephrine. And so that can increase the force of contraction and the duration of stimulation. Some vocabulary here. Power is the maximum amount of tension that can be produced in a muscle. Endurance is the amount of time that an activity can be sustained. So power, if you're looking for just pure power, that would be like the 100-yard dash, right? You're full out as fast as you can, as powerful as you can. Endurance would be you're running a marathon. So you can maintain this activity for 26.2 miles. Both power and endurance depend on what type of muscle fibers are being utilized and your physical conditioning. So the more conditioned you are, the more you exercise, the greater your power and the greater your endurance. We have three types of skeletal muscle fibers. Fast fibers, which contract very quickly. They are large, they have large glycogen reserves, but very few mitochondria. So they have strong contractions, but they fatigue quickly because the mitochondria is where we produce our ATP. So if you don't have a lot of mitochondria, you cannot perform the aerobic respiration. And so it's very hard to create additional ATP. So you're going to have quick fatigue. Slow fibers are slow to contract, but they're also slow to fatigue. So you're going to last longer when you're utilizing slow fibers. They have a small diameter with more mitochondria. So they have a high oxygen supply, so they have more mitochondria, and they contain myoglobin, which is a red pigment similar to hemoglobin in the blood, and myoglobin binds oxygen, which gives them a greater oxygen reserve. They're about half the size of fast fibers. And then our intermediate fibers are exactly what they sound. They are mid-sized. They do have myoglobin, so they do have oxygen. They have more capillaries than fast fibers, and they're slower to fatigue than fast fibers. So they're kind of middle of the road fibers. If we look at fast fibers versus slow fibers, here you can see the slow fibers have a smaller diameter, they're darker in color because they have that myoglobin, and they're more fatigue resistant. The fast fibers, which are shown down at the bottom here, those are lighter in color, they are larger, and they're more easily fatigued. Our white muscle is mostly fast fibers, very pale muscle fibers, and so those are like a chicken breast. They're used for very short bursts, but they are not sustainable using those muscles. Red muscle is mostly slow fibers. They're darker, like chicken legs. You picture a chicken is walking around on their legs all day long, so they need to be able to sustain the contraction of their legs. Most human muscles are mixed fibers, and so they're more pink in color. And that I can attest to after cadaver dissection for a year. Muscle hypertrophy is where your muscles are growing in size from heavy strength training. So we increase the diameter of muscle, we increase the number of myofibrils, and increase the number of mitochondria as well as the glycogen reserves. So everything about a bigger muscle, the muscle is going to work better and last longer. Muscles also can atrophy through lack of activity. We reduce our size of our muscle, reduce the muscle tone, and reduce the power that we're able to generate. Physical conditioning improves both power and endurance. Anaerobic activities like weightlifting or the 50-meter dash use your fast fibers, and you fatigue more quickly with strenuous activity. You can't sustain it as long as you can sustain aerobic activities. You can improve your anaerobic activity by frequent, brief, intense workouts and hypertrophy. You're going to increase your muscle size, and so you're going to increase your strength and your like speed in a 50-meter dash. Aerobic activities are, you can do them for a longer period of time. They're supported by mitochondria using oxygen, so they require oxygen and additional nutrients. You can improve your aerobic activity abilities through repeated training and through cardiovascular training. The most important thing about physical conditioning is regular exercise is the most important. And it's good to have a mix between aerobic and anaerobic activities. Aerobic activities are great, but they're not really building muscle as much as anaerobic activities. Building muscle is going to increase your endurance and your overall strength and increase your base metabolism. Aerobic is, but also a really good cardiovascular workout, which is going to help your system overall. So mixing it up a little bit is the most important thing, and just getting out there and exercising. When you're inactive, you lose muscle. So your muscle tone indicates the sort of base activity of motor units and skeletal muscles. So if you are nice and buff and strong, that means that your motor units are working really well. If you are inactive, your muscles are going to become untoned, they'll decondition, become flaccid, and the muscle fibers are going to break down. They're going to become smaller and weaker. With prolonged inactivity, so people who are inactive, like say bedridden, their muscle fibers can start to get replaced with fibrous tissue, and they just don't function like they once did. So that's the end of our cardiac muscle. Like I said, we spent 24 pages on cardiac muscle, and we're going to spend the next, 6 or 7 pages on the other types of muscle. So let's move on to cardiac muscle. Cardiac muscle is made up of muscle cells called cardiocytes, which are cardiac muscle cells. They are small. They have a single nucleus, unlike skeletal muscle, which are multinucleate. They have short, wide T-tubules, but no triads, which means the sarcoplasmic reticulum do not have terminal cisterns. They are aerobic. They have a lot of myoglobin and mitochondria, so they have a lot of oxygen and use aerobic metabolism. And the cells are connected at intercalated discs. You remember those intercalated discs are gap junctions, which rapidly transport ions from one cell to the next and allow these cells to have a coordinated contraction cycle. So here we go. Intercalated discs are the contact points between cardiocytes. They are gap junctions. They help maintain the structure, enhance molecular and electrical connections, and conduct action potentials from one cardiocyte to the next. The cardiocytes are coordinated in contraction because they are linked by those intercalated discs. So it makes them work as a mechanically, chemically, and electrically singular unit. Here's our picture of our cardiac muscle tissue. So if you're looking under a microscope, which this week you do want to look up microscope slides of cardiac muscle, skeletal muscle, and smooth muscle so that you can tell them apart. But on this one, I'll use blue. Right there is showing you an intercalated disc, and that is where those muscle cells are going to connect together. This is the end of the intercalated disc shown here on that one. Looking down at this bottom one, you can see that the sarcoplasmic reticulum is this sort of spiderweb region here, but it is missing those thicker bands of the cistern, which you would normally find on either side of the T-tubule in a skeletal muscle. This is just another picture where you're looking at that T-tubule and the missing triad with the no terminal cistern. Functions of cardiac muscle tissue. Automaticity, which means that the cardiac muscle tissue can actually contract on its own without interference from the central nervous system. It's controlled by pacemaker cells. You're going to learn a lot more about that in A&P2. It's important to know that even though it can function without neural stimulation, it does always have sympathetic and parasympathetic input, but in the absence of sympathetic and parasympathetic input, the heart would continue to beat. So that's what automaticity means. There's a variable contraction tension, which is controlled by the nervous system, which means the nervous system can make your heart pump harder and faster, or less hard and slower. It has an extended contraction time, so the contraction of cardiac muscle is 10 times longer than the contraction of skeletal muscle, and it prevents wave summation and tetanic contractions because it has a very long refractory period. So the refractory period is the after the contraction, before additional stimulus, a time when it cannot actually create a second contraction. And so when that refractory period is long enough for the cell to come back to normal before its additional stimulation, it prevents tetanic contractions. And that's very important because you don't ever want your heart to go into tetanus. If your heart stops beating, all of your tissues are going to start to die within seconds. So now we're going to move on to smooth muscle, which is our last bit of muscle tissue. Smooth muscle forms around other body tissues. So it's in the walls of blood vessels and helps to regulate blood pressure and flow through our arterioles. In the reproductive and glandular systems, it assists with glandular secretion and the movement of materials through our reproductive system. In the digestive and urinary systems, it forms sphincters, which the sphincters in your digestive and urinary system give you the ability to, well, those are more of a skeletal muscle sphincter that gives you the ability to control urination and defecation. But there are also internal sphincters, like between the stomach and the duodenum, which is the beginning of the small intestine, that keeps the contents of those two separate until the stomach contents are processed enough to be able to safely move on to the duodenum. And so that through the digestive system becomes very important. And in the integumentary system, we already learned about the erector pili muscle, which attaches down into the base of the hair follicle, and when contracted causes your hairs to stand up on end. It gives you goosebumps. Those are actually smooth muscles. You do not have any control over them. Smooth muscles are non-striated. That's what makes them smooth. And they have a different internal organization of actin and myosin. They don't have sarcomeres, so they don't have the actin and myosin overlapping one another, but they have this sort of crisscross pattern of actin and myosin. And so when you can see when the muscle is contracted up here, the actin and myosin are kind of spread out. And they connect at what are called dense bodies there. And then when it contracts, it pulls that whole muscle smaller in every direction instead of just shortening the muscle fiber like a skeletal muscle does. Characteristics of smooth muscle: they are long, slender, and spindle-shaped cells. So spindle-shaped means they're wider in the middle and tapered on either end. They have a single central nucleus, so they are uninucleate, like cardiac muscle cells. They have no T-tubules, myofibrils, or sarcomeres. They have no tendons or aponeurosis. They have scattered myosin filaments instead of myosin filaments lined up nicely. The myosin fibers have more heads per thick filament than skeletal muscle does. They have thin filaments attached to those dense bodies, and the dense bodies are what transmit the contractions from cell to cell. They are also involuntary. Cardiac muscle is involuntary. Skeletal muscle is voluntary. Traditionally, I would make a list on the board for you, like a table that has skeletal muscle, cardiac muscle, and smooth muscle, whether or not they are uninucleate or multinucleate, if they are voluntary or involuntary, and what's the other one? There's a third thing I usually put on that list. So I would go over all this and figure out that third thing and make yourself a chart so that you can really keep the characteristics of each different type of muscle separated. For contraction in smooth muscle, we have free calcium in the cytoplasm, which triggers contraction. It binds with calmodulin in the sarcoplasm and activates this enzyme called myosin light chain kinase that breaks down ATP and initiates the contraction. On this slide, I would know that calcium binds with calmodulin in smooth muscle tissue. Functionally, the thin and thick filaments are scattered, so the resting length is not related to the ability to develop tension because there's not that that crossover like we have in skeletal muscle where we've got minimal zone of overlap is harder to create a contraction. Maximal overlap, there's very little room for contraction. So in smooth muscle, it can function over a wide range of lengths because it is not dependent on the actual overlap of actin and myosin. So that's a really important feature especially in organs like the stomach that needs to expand and then contract, and in the uterus which needs to expand and contract. So the smooth muscle can function in the expanded state as well as in that contracted state. We've got multi-unit smooth muscle cells connected to motor neurons, and visceral smooth muscle cells are not connected to motor neurons, but they are controlled by pace setter cells. So cells within like the digestive system can control their own contractions and propel food and digestive boluses through the digestive system, and those can be acted on by local hormones and other sort of things that get released locally instead of requiring a motor nerve innervation. So I really struggled on that slide. Smooth muscle tone maintains normal levels of activity and is modified by neural, hormonal, and chemical factors. So that was what I was just discussing in the digestive system. Nerves can force them to contract, hormones can force them to contract, and different chemicals locally can also force contraction. So that is the end, finally, of our muscle lecture. This traditionally is the most challenging lecture, so please, if you have questions or need additional clarification, you can email me or wait until our Zoom meeting if you're in a course where we're doing Zoom meetings, and I'd be more than happy to give you additional information about any of these topics. But for now, have a great week, and next week we are going to learn every muscle in the body. So you have something to look forward to.